only 1% of your DNA codes for making proteins, so 1% only determines who you are. What about the other 99%? The other 99% is simply junk DNA, nada, trash, or as called by scientists, non-coding regions. Surprising, right? Well, this is just the beginning of the story, or the story believed up to 2012. The term junk DNA was used by scientists to describe the regions of the DNA that do not code for proteins, as it was believed that these regions had no purpose. However, some of these regions we discovered to be related to the control of gene activity. An article was released by Time in 2012 that indicated that junk DNA is not so useless after all. Scientists found that the DNA is a lot more complex than we ever thought, and these non-coding regions are active and essential to the activity of the DNA. Yet, the function of many of these regions is still unknown. Some of these non-coding regions include regulators of gene expression, introns, telomeres, genes for tRNAs, tandem repeats. Let's talk about each of these regions. Regulators of gene expression. There are different regulators of gene expression. We have mentioned one type before, promoters. Promoters initiate DNA transcription. They are short regions of DNA that are found just ahead of the gene on the DNA strand. The enzyme RNA polymerase binds to the promoter to initiate transcription. In order for the promoter to be activated, specific binding proteins bind to a region close to the promoter called enhancer. Enhancers are binding sites for these proteins. They could be found before or after the gene on the DNA strands, sometimes before the promoter. So both promoters and enhancers play a role in activating DNA transcription. Other regions on the DNA strand that act oppositely to enhancers are called silencers. They provide binding sites for specific proteins that prevent RNA polymerase from binding to the promoter site, so they prevent transcription. Introns are other non-coding regions of DNA. We have mentioned before that introns are only found in eukaryotes' DNA and not in prokaryotes, and they must be removed immediately from mRNA after DNA transcription is completed and before translation is carried out in a process called splicing mRNA. The function of these areas is not yet confirmed, but some scientists believe that these regions have a role in regulating gene expression. Telomeres are also non-coding regions that are only found in eukaryotes. As you see, they are found on the ends of eukaryotic chromosomes. They make up around 5 to 60% of the genome of eukaryotes. They are high repetitive sequences that were believed once to have no role. However, today's scientists believe that they protect the DNA during the replication process. They believe that these areas prevent the loss of genes near the end of the chromosomes during DNA replication. There are non-coding regions of the DNA that play a role in making our DNA unique. These are called tandem repeats. A tandem repeat is a sequence of DNA base pairs that is repeated several times on our DNA strand, where the repeats are next to each other. The number of times these areas are repeated varies from one person to another. These tandem repeats are referred to as short tandem repeats, STRs, also known as variable tandem repeats. For example, in this short tandem repeat, the base sequence CATA or CATA is repeated 6 times for one individual. In another individual, this tandem repeat could occur only 3 times. In crime scene investigations and in paternity tests, tandem repeats could be used in identifying the DNA of the suspect or the father. This could be done by using gel electrophoresis and DNA profiling, which we will discuss later. We know so much already about DNA, yet DNA is still a puzzle that has not been completed yet. Gene expression is a very complex matter, and if we discover all the factors that control gene expression in all cells, maybe one day we can understand all about diseases and solve the puzzle of DNA. See you next time.